So I'm Damian Green. I'm a physician scientist at the Fred Hutchinson Cancer Research Center and Seattle Cancer Care Alliance, where I run a research laboratory doing translational work, which we bring to patients. The reason I'm at the Fred Hutch is because we have an outstanding system for actually being able to develop things in the laboratory and bring it to patients in real time, and that's why I do what I do. So one of the areas in myeloma that I frankly think needs to be accelerated is in the area of improving conditioning therapy for an autologous stem cell transplant. So, as you know, we've seen just remarkable advances, dramatic advances in the management of this disease, resulting in more than doubling, tripling of improvement in survival for patients. However, there's been no change to the conditioning regimen for autologous stem cell transplant really since the beginning. It's high-dose melphalan, that's what's being given now, and that's what's been given in the past. A few centers have dabbled in other approaches, but that is the standard of care. And I think we can do better. As we learn more about the disease, we can take advantage of the discovery with respect to the immune system and then use that as part of a conditioning regimen. So for many years in my laboratory, we've been focused on targeting CD38. It's really gratifying for me to see all the excitement about CD38 antibodies that both at ASH this year, ASH last year, the approval from the FDA just last year. But I don't think that CD38 antibodies alone, and I think it's been proven by the data, are the final punch that's going to take out this disease. They do, however, represent a target on the surface of those cells that we can then harness and bring to the myeloma cells an extra punch. There are a number of ways of doing that. Some people attach different drugs to attack those cells, and that's an important approach. We have at the center from the early days done pioneering work, I would say, from my mentor Ali Press and others at the Fred Hutch, developing radioimmunotherapy for different forms of cancer, and my focus has been doing that for myeloma. That's why we targeted CD38 years ago, but now, after many years of research in this area, we are on the cusp of having a clinical trial for patients. And in that trial, what we're going to do is we're going to take melphalan as the backbone. We're not going to take that away, at least for now. Ultimately, I'd like to take it away. But for now, we'll use that as a backbone, still give patients melphalan, but add to that approach CD38 antibodies that bring with them a small radioactive particle called an alpha emitter. And alpha emitters are particularly attractive to us because they carry with them a very high energy, and yet they deliver it over a very short distance. So if you put a CD38 antibody onto a myeloma cell and it's carrying along with it these alpha emitters, they can't go through a piece of paper, which means if you target them to the cells, the chances of their damaging other cells is very, very low, and we've demonstrated that in the research setting. In that context, for the treatment of myeloma patients, when we deliver it, we will bring that whopping punch to the surface of the myeloma cells and deliver it to them directly, the radiation, which it actually only takes about 5 CD38 antibodies bound to a myeloma cell to kill that cell, and irreparably, because it causes double-strand breaks in the DNA and the cells cannot repair themselves from that cell injury. So we have worked in the laboratory, again, for years to develop the right approach for this delivery mechanism, and we're now at a point where we anticipate opening a clinical trial using that approach to condition for transplant within 12 to 14 months. Will depend on FDA approvals and other elements, but trying to do that as fast as we possibly can. And I believe that that is harnessing the advances in immunotherapy with the benefits of a stem cell transplant. And our idea is to use this approach to target every last one of those myeloma cells and get rid of them. You know, there's a lot of excitement about minimal residual disease. Our expectation would be knock the patient's disease down with an induction form of therapy, and then once you have the disease knocked to a very low level, when you give the melphalan conditioning, add the alpha emitter conjugated CD38 antibody, which our design is to eliminate MRD using that approach and then rescue with the stem cells afterwards. So it's at a point which is very promising, and we will have a study open for that soon.